So hi everybody and welcome to our workshop for the Soil CRC's Northern Community of Practice for on soil constraints. So I'd like to welcome Esan Tabakali from New South Wales DPI and Maron from Griffith University, who's going to be each talking to you today, covering a couple of topics related to soil constraints. That's okay. Richard's just sent me a message. So as with the other webinars that we've had, please use the chat box so that if you've got any questions along the way, and Esan's happy to take questions along the way as well. I'll post a few things in the chat box, so keep an eye on that. And yeah, take notes if you like. And like I said, we're recording it. We'll be able to have a look back on it afterwards. Has anyone got any questions before we start, or any queries? No, you're all happy. So yeah, anyway, if you have, yeah, just post into the chat and I'll keep an eye on that for Esan. So thank you, Esan, for making yourself available today to talk to the community of practice for Soil CRC, and I will hand it over to you. Thank you.
Thank you, Felicity. So everyone can see my slides?
Yeah, so again, we're looking at the presentation one where we can see the two screens.
Yep, okay. So just quickly, I stop sharing this.
Yep, that's great. If you just make it a presentation
screen. Yep. Easier, one sec. How about now?
Beautiful. Thank you.
Is it fixed, Felicity?
Yes, it's all good.
Good, okay. All right, so good morning, everybody, and I trust everyone can hear me now. Yep, great. So I thought what I'm going to do today, so based on the information Felicity gave me, I saw a major interest on soil and subsoil constraints, and basically this was about sodic and saline salt and acidic salts. So for the first maybe 20 minutes, I'll talk about saline and sodic salts. What are they? How they actually form in agriculture. My research is mainly focused on broadacre cropping system, but the concept of sodic saline salts, their amelioration and management is pretty much the same for either you're in the sugarcane industry or horticulture and so on. So I think if you understand the concept and what is the problem, then we can find a solution for it in different farming system. So very quickly, I just want to talk about the distinguish between a saline salt and a sodic salt, because this is what is mostly confused even in science these days. I use the broader concept of salt affected soil, and under that umbrella of salt affected soils, we have so many different categories. A soil can be saline, sodic, it can be saline and sodic, and then you have a complications of pH influence there, which makes that management of these soils also very difficult, because it's not just one single problem, it's a number of different issues that needs to be tackled at the same time. So basically, if you look at the left-hand side, in this image that I put and written a saline soil. A saline soil is saline when you have soluble salt in the soil solution. Okay, so sodium chloride or different forms of salt. It doesn't need to be either sodium or chloride all the time. It can be, you know, calcium, magnesium, and different carbonates in the soil. When you have them in soluble form in the soil solution, the soil is becoming saline. And when that soil is leached by rain or irrigation over a number of decades, some of that, some component of that salt can leach down into the deeper soil profile. But in this case, when you see in the sodic soil, the sodium can remain within that clay aggregate. And in the next couple of slides, I'll explain this in more details. But when you leach out that salt and the sodium stays in the clay, the soil is becoming sodic. So that is no longer a soluble salt, but it's a salt that is incorporated into different plates of the clay in your soil. And their problem and how the crop feels the pain is completely different. So I just wanted to set that difference between a saline and sodic soils, and from now on, I focus on what are the differences. Just imagine if you have a clay particle and that brown large circle on the left-hand side. That clay particle is usually negatively charged. So, and for this clay to bind together, you need to have a positive bridging between the clay particles to make that binding, okay? And that's when you have calcium, magnesium, sodium, and different cations that form that bridges. Each of these elements have a diameter and a strength, atomic strength, that making the binding. And if you, for example, remember when we were kids and grown up, our moms were encouraging us to drink more milk because of the calcium in milk. The calcium plays, for the bone structure, calcium plays a very similar role in clay particle. You want more calcium on the clay. This is not in the soluble form, this is on the exchange side. You want that calcium on the clay to have a better structure of the soil. And in comparison, when your clay is dominating with more of this sodium, which is a lot smaller molecules, then the bondings are a lot weaker. And when they are weaker, the soil is not stable. That's when you have a structural issue. What is the issue in a sodic soil? On the left-hand side, on the right-hand side, you see a petri dish where there is a brown liquid. That is a clay that is in contact with water. Because that bonding, those sodium, too much sodium in the soil, the bondings are weak. The clay, which in a normal situation should be stable, is actually slaking and dispersing in the soil, and you have colloidal dispersion in the solution, which can cause problem. What's the problem? These are tiny, tiny colloidal particles that at the landscape, if you have many hectares of this sodic soil, what happens is that as the soil dries out after the dispersion happens, those fine particles can sit like a cement. That's where you get surface crusting or blockage of the soil pores. Imagine if the rain or irrigation water is going to penetrate or infiltrate into the deeper soil profile, and you get waterlogging. The reason for that is that this cementation or hard or the surface which is dispersed clay after it dries out, preventing that infiltrations to happen. So in a sodic soil, we often have a major soil structural problem. I want to simplify this for you further. So just imagine on the right and left hand side, you have two clays. Yeah. So it's just a bit of soil in your hand. As I mentioned, these are negatively charged, and they need to be connected to each other to form a structure that is your soil, and for the crop to go through it and see water and nutrients. In the middle, you need a positive charge to bring this together, right? So you need a positive charge to bring the two negative soil clay together, and the cation that does that can be calcium, potassium, sodium, or magnesium. When you have a large amount of sodium as that positive charge, you have a sodic soil relative to your other cations such as calcium, magnesium, potassium, which are stronger in bonding. And that's where the problem happens. Why? Because the proportion of that sodium on the soil particle is basically providing that really weak bonding of soil aggregates, and you need to add an amendment such as gypsum, which is a very common product in agriculture, to compensate the amount of calcium that you needed to have on that soil, and it's not there anymore, for the sodium to come off the clay, so it's an exchange, for the sodium to come off and replace it with calcium so you can improve the soil structure. So this is the concept of a sodic soil, and really if anyone has a question now, I think it's the best timing to answer, because I really want that concept to be spot on in our understanding. So there is no salinity issue in this example. It's just the soil which is sodic, and it has a structural problem. Salinity is more a chemical problem that I will explain it later. So again, a little bit of more schematic presentation of what the problem is. On the left-hand side, you have a dispersed clay, because now now this clay is hydrated and there is no structure, whereas when you have flocculation, which is the term opposite to dispersion, those clay particles that are well structured because there is enough calcium and organic matter in the system, they stay together and form a soil structure. When the rain or irrigation comes through, on the right-hand side, the water will easily go through the soil pores, and the roots also will go into the deeper soil profile. However, when you have dispersed clay, you get that waterlogging events in this case, and the water infiltration or soil hydraulic conductivity is a lot more reduced. So this is the problem that we want to improve, which is a lot actually more difficult than many other constraints in agriculture. So how to identify this in the field? Yeah, I think the first step is very simple. If you think you have a sodicity problem, and by the way, what I really need to mention right at the beginning is that the term sodicity is a very general, arbitrary definition of the problem. Dispersion is really what we are after. We want to identify if our soil disperses or it doesn't disperse, because you may have a situation that according to your soil test, your soil is sodic. However, if that sodic soil doesn't disperse, which it can happen, it won't respond to gypsum. That's when a lot of people ask me, oh, okay, so based on the soil test, my farm is quite sodic, but there is no benefits of gypsum application. In most of the cases, that happens because the soil, even though it came as dispersive, it wasn't actually, the soil was sodic, it wasn't actually dispersing, and it is the dispersive nature of the soil that is responding to gypsum application. So in the field, if you want to identify it, it is very simple. You just need a few petri dish or even a plastic jar or a cup, and you need clean water, and you need a few aggregates of your soil that you suspect it has a problem from different depths and across the paddock. All right, so because it's very variable, you need to have a good understanding of the variability of this problem. You put the aggregates into the cup, you add the clean water, and after an hour, if that cloudiness, that milky cloudiness around the particle appears, then you know you have dispersion in the soil. Whereas, as you can see, those aggregates that sit there in the petri dish intact, they are not actually, they won't need any gypsum application. And that's the problem, because most of our dispersion these days happening in the subsoil, according to these different depths, 0 to 10, 10 to 20, 20 to 40, you will see that the topsoil is actually not dispersive, and it is the subsoil that has that problem. And if you leave or apply your gypsum in the topsoil, you still have the problem in the subsurface, in the subsoil, and you didn't have any gypsum requirement for the topsoils anyway. So that's where you need to be having a good idea of what is the problem. After you identify you have dispersion, then to do a soil test, because that soil test in the lab will inform us how much gypsum is required to be applied into that soil. The first Petri dish test is just observation. The second soil test is to give you the prescription of how much of what material is likely going to fix the problem. I'll just give you some other example. This is just different soil types from southern part of Australia, Wagga, South Australia, Victoria, in those long columns that you see. Simply, I just mix 100 gram with a few hundred mils of water. You will see the first 4 are quite dispersive, so you have that cloudy, milky solution after we mix the soil and water together. And the right hand side column is very clear. However, that soil test was actually 40% sodicity there. The reason for that soil that is sodic but not really dispersing anymore is because that soil at the same time is very high in salt content. And that salinity, most of our saline soil has a good structure. When you have a saline and sodic soil, the salt concentration fluctuate that amount. It's like a gypsum. Gypsum is a salt you add it to the system, so that dispersion actually reduces. And if you just go based on this an ESP test at the first place and apply gypsum, only the first 4 soil will respond to gypsum application, and the last one doesn't need any gypsum application because it's not dispersive. Those smaller test tubes on the right hand side shows what gypsum does to a dispersive soil. So starting from a sodic dispersive soils from the left hand side all the way to the right hand side, we increase the amount of gypsum addition. And what happens, as you can see, the amount of dispersion is actually settling down. And this sort of test can tell us at what concentrations of gypsum we actually fix the total amount of that dispersion. And we can also, based on these studies, decide how much gypsum is needed to be added in the field. Okay, so from these first few slides, I wanted to set the point of what sodic soil is, how we test it in the field, what needs to be done after that in the lab, and what is actually the effect of gypsum when people say we need to add gypsum into the soil. We need to reduce that dispersion in the field.
We've got a couple of questions.
So this was a very simple overview of the problem in the field. However, if you think about what are the soil or subsoil components in our farming systems, I can categorize them into 2 categories. Chemical constraints. We have acidity, sodicity, alkalinity, because high pH itself is always a problem, and also salinity. And we have compaction that Miran will talk about it. And there are waterlogging issues that are interrelated to acidic soils or soils with a poor structure. Because if you have that dense clay, you get transient waterlogging, which is a physical problem that is associated with the chemistry, which is that sodicity and high dispersion level. So this is the simple way. However, the next graph will make everything more complicated. This is the reality. This is what happens in our farming systems every day. Because if I want to take into account, each of these soils can have 3 different pHs. They can be acidic, neutral, or alkaline. And you have a non-salt affected soils on the bottom left-hand side. And then you have a saline soils or a saline sodic soils or a sodic soils. And what you need to do, or all of us we need to do in our farming system, is to understand where in this chart we are sitting. Do we have a sodic soils with a pH of less than 6? Oops, with a pH of more than 8? Because if the pH of an sodic soils with an alkaline pH more than 8 is our case, which is actually the case for most of our farming system, we have a few issues. We have soil structural issue. We have that dispersion. We have waterlogging every now and again. We have carbonate and bicarbonate toxicity because that alkaline soils are characterized by high carbonate toxicities. And you can also get some sort of deficiencies of nutrients. And just adding gypsum into these soils is not going to fix the problem. So I call that a multiple subsoil or soil constraints, which we need to correctly characterize what the issues are and what depths can that be achieved. And if we can manage the soil to achieve productivity improvements, then we can go ahead. Otherwise, it is very risky. Soil management is expensive, and we need to make decisions based on minimizing risk and return in investment. So the point of this graph or this diagram is to just highlight what a complex problem is subsoil and soil constraints when you interrelate the pH, salinity, acidicity, and I think at some stage I came up with 16 different categories of these soils, but we can actually shorten them or summarize them into some really key ones, and I'd like to focus on alkaline sodic soils for the rest of this presentation, and into this, and also the alkaline saline soils. Most of our acidic soils, you don't need to be really worried about sodicity, because the pH, the high pH in sodic soils is one of the reasons that we get that dispersion. Okay, so acidic soils in comparison to these are less, they have less chance of dispersion. So if I want to quickly talk about distribution of sodic soils in Australia, of course, there is a wide range of cropping systems all the way from the northern region to the southern region and western region that is that have this problem of sodic or subsoil or topsoil. And then in terms of grains industry, the yield gap associated with the soil constraints is basically anywhere from 0, where there is no problem, up to 0.4 ton per hectare per year. So if I add all those values based on the recent study, every year we are losing about $1,300 million per annum just because of the sodicity issue in Australia across the whole northern, southern, and the western region. And so it's a significant figure in our yield gap. So this is the potential that we can achieve, and we don't achieve it because sodicity is there. What is the major problem in sodic soils and acidic soils, or wherever you have a soil constraints problem? You can have a waterlogged system, you can have a crop establishment issue. There is one factor in common between all these problems, and that is the poor water use efficiency. The concept of the work you want to do and you want to develop a management for is to improve the water use efficiency of these systems, because if you imagine you have a profile of water that your crop should effectively dry that out from, so this is your soil profile at the beginning of the season. I call it the upper limit and the lower limit. If the plant have access to this profile, you will have a perfect yield at the end of the season. However, when you have a subsoil constraint in your system, that bucket size is much reduced. Okay, what I like to do in our work with the subsoil constraint is to improve the bucket size, or the plant available water, which is potentially available but the crop can't see it because of that soil or subsoil constraint. There are a few options to achieve this. We can achieve it by soil management, and you heard probably about some of our work on subsoil manuring, or adding organic amendments, or combination of organic and inorganic amendments into the subsoil to improve that poorly structured environment. We can improve our genetic tolerance. We can come up with new varieties that are better performing in some of these hostile conditions. And the third option is to make new amendments. Obviously, lime and gypsum has been used in Australia since 1920s, and we are still doing research on the same product. One of the reasons that we haven't achieved a lot of breakthrough is that these conventional mined products are not highly effective in ameliorating the subsoil problem. They are good for the topsoil, but they don't really influence the subsoil effectively. So as part of the CRC project, we are actually focusing on the subsoil management, so soil management issues. This is the program 4 under Lucas's leadership. And then on the right-hand side, we are also improving the formulation of the new amendments. Can we improve the mobility of gypsum and lime to penetrate into the deeper soil profile to improve the soil environment? So just I want to give you some encouragement. This is what excites me all the time. On the left-hand side, you see a subsoil from Victoria that is quite poorly structured, dense clay. And after 4 years of adding organic amendments into the soil, you come up with a subsoil which is a lot more well aggregated and transport unit structure. So there are quite a lot of potential there. And these changes in the soil structure has been associated with up to 70% increase in yield for up to 10 years, right? So there is like often you need a one-off application and the effect lasts for a number of years, depends on the environment and depends on what you're adding into your soil. I'll give you some case study. I think I'm running out of time and I've got quite a lot of slides to go through, but I'll go very quickly. Felicity. So this is an example from southern New South Wales. Okay, so we've got a property that has a pH of about 5 in the surface, and as you go into the deeper soil profile, that pH goes up to 9. So this is a classic, actually sodic dispersive soils that even though the pH in the surface can be neutralized, as you go into the deeper soil profile, the pH increases by depth into extreme alkaline condition. And that pH itself can cause a problem because of that carbonate and bicarbonate toxicity. Then the ESP, which is your exchangeable sodium percentage. This is an indication of how sodic your soil is. And in Australia, anything above 6% is sodic, okay? You can see that we have a non-sodic topsoil, and as you again increase, as you go down to the soil profile, the sodicity or dispersion increases up to 30–35%, and this is what I'm talking about. So most of our topsoils can be either looked after or they are not actually sodic or alkaline. So it is the subsoil that is my interest, and that ESP itself has problem with poor structure, waterlogging, and some dispersion. And salinity here, which is electrical conductivity. Again, in Australia, anything above 4 considered saline. It's about 6 or 7 when it hurts the crop, like wheat and barley. And basically, you have osmotic stress or some sort of sodium and chloride toxicity symptoms. And as you also can see in these Petri dishes, the topsoil is not dispersive. As you increase the depths, it can be dispersive. Okay, so what we've done here in this case, we tried a number of different amendments. We wanted to fix the subsoil, okay? So the topsoil, even though it's okay, most of the farmers would apply the gypsum to increase their, assuming they increase the productivity, but if the soil is not dispersive, no gypsum or no amendment is required. It was my interest to see what are the possibility to fix that subsoil. And quickly I run you through some of the treatment we use. Of course, gypsum is one of the most commonly used amendments in agriculture for amending a sodic soil. In addition to that, I wanted to see if organic amendments, a manure or a plant biomass, this can be as simple as a wheat stubble or legume stubble after your harvest, because it's very rich in carbon. Can we incorporate that into the subsoil and let the microbial decomposition of that stubble to help the soil structure by adding the carbon and some nutrients in that subsoil environment, which is poorly structured. Because in sodic soil, we have a structural issue that the structure needs carbon to bind the aggregates together, in addition to gypsum that fix the dispersion. So we have actually a couple of issues here. So we had here a number of different treatments, from manure, from pea straw, from just nutrient itself, because the subsoil is quite infertile, and we wanted to see what are the possibility to to make changes in this hostile subsoil environment. We used a machinery, a deep repair that is fabricated and designed in DPI New South Wales. With this machinery, we can apply organic amendments up to 20 tons per hectare. We can also add lime or gypsum from this box, and then in front of this hopper, there is a liquid tank that we can apply liquid fertilizer if we want to at the same time, or as just on their own application. Okay, so so this is the machinery that we used, and it has 5 tines, and the rip lines are about 50 centimeters apart from each other. This is an aerial photo of the trial in 2019. Remember, this experiment was set up in 2017. We had a barley crop in the first year, then we went to a wheat crop in 2018, and then we went to canola in 2019, and now we are back into wheat in 2020. Very quickly, I just run you through the data from 2019 season that some of those amendments, such as deep manure, deep gypsum, and deep P-hay, which was a pelletized product, plus NPK, resulted up to 36% increase in yield. This is the absolute yield relative. These numbers on top of each bar is relative to control, which is on the left-hand side. So what I really like to use my time wisely, because I know I don't have much, is to actually explain what happens after you add some of these amendments into the soil. Before going to that explanation, I also wanted to give you some idea of what happens in the second season in 2018, which was a very dry season, if you remember, at least for New South Wales and southern parts of Australia. Again, some of those deep applied treatments, which is a combination of organic amendments and not necessarily manure. This is just simple P-hay plus gypsum plus NPK, resulted up to 53% increase in yield. So the message here is there are potentials or significant potential to improve productivity if you are managing your subsoil. And it's a one-off application there that you need to choose wisely what you need to add. So first thing that you are looking for in this environment is whether we change the bucket size, and that concept of the plant available water. What is going to affect the bucket size is the soil structure, and also the amount of root that is now going to be in the subsoil affected by this management. If you don't have more root to take up more water, you cannot have more biomass and you cannot have more granules. So we do some tests in the field looking for the root accumulation in the subsoils affected by different amendments. If you look at control on the left-hand side and some of the treatments on the right-hand side, you will see we have a significant increase in the amount of root in that depth of 20 to 40 centimeters after 3 years of placing the organic amendments into the subsoil. Okay, so that extra amount of root there means they have more access to the water, and if the water is going to be there, okay, so that's the trick. You need to have a remaining water into the subsoil, and you need to improve the soil structure to release that water, because if you don't release it from a dense clay, even the root is there, you won't still going to see that. So in our root studies, we have a strong evidence at the moment over a number of years that we have a significant amount of extra root, and we also have evidence that we have a much better hydraulic conductivity in our soils, and the hydraulic conductivity is the capacity of the rain to penetrate to the deeper soil profile, because you want to improve that waterlogging condition if you experience it. Even then, you still may have water storage issues. So we do in-field hydraulic conductivity studies, and very similar to our observation with the roots, you will see that the deep placement of organic amendments improve the hydraulic conductivities quite significantly over a number of years. So that means you actually store or improve that dense clay to let more water to go into the subsoil. Okay, and this measurement here is how much water is being taken up by the plant. Okay, Don't worry about the complexity of this graph. Just focus on, please, on the fact that if we are shifting to the left-hand side of this graph, the plant is taking more water from the subsoil. So these two bars are control or deep nutrients, where we just put nutrient into the subsoil, compared to doing nothing. And you will see that both of them are quite similar in this and in the lower soil profile. When you start looking at deep organic amendments that we left into the subsoil, you will see that there is a shift. About 10 to 15% are calculated across all these treatments. There's about 10 to 15% extra subsoil water is being used, and that is an indication that we improved the soil structure and the crop had the ability to take up that water. And a back of envelope calculation there is for an extra 10 mils of water, if you make that available and the crop has access to it, that's about 0.5 ton per hectare of extra grain yield. Okay, so that's the conversion based on modeling. And if I want to summarize that, what happened to the soil that we can see these differences, if you just look at the pH, remember the soil had a pH of about 9. When you add gypsum to this soil, the gypsum is reducing the pH quite significantly. So what you want to achieve here is to get rid of all the toxicity of the carbonate bicarbonate in this region, and by reducing the pH, you're also improving the dispersion. Then you add organic matter, but organic matter doesn't do anything with the pH. It's just the gypsum effect that is making an influence there. In terms of the sodicity, you also see a very little reduction in the soil ESP by adding gypsum. Again, organic matter doesn't have a major influence. So far from the chemistry point of view, we are just dealing with the effect of gypsum. In biology, however, when we measure carbon in the soil, you will see it's exactly the opposite trend. Gypsum doesn't play a role there because there is no carbon in gypsum, but it's the organic matter that has a huge amount of carbon that in this case is actually in the soil fraction. So this is a measurement of how much carbon is in different soil fraction, and that carbon can be a glue for improving the soil structure. If I measure the soil structure in the field, you will see a 4-fold improvement from the control to some of our best performing treatments. And we started to see in the field some biological activities and deeper root penetration, infiltration in these conditions, which is quite encouraging because if you look at on the right and left hand side of this rip line where the amendment is, there is no activity or there is no presence of root. It is where the amendment is being placed. And that's a strong message because there are windows of opportunity here to fix or to improve, not to fix, to improve some of these hostile conditions. So, and some more visual observation. This was taken in September 2018, which was a very, very dry spring. And it's a comparison between your control on the right and ameliorated area on the left-hand side. And I just took a few crops from each of these locations, and you can see the vigor and the health of the crops are quite significantly different from each other. Again, more encouragement on what can be achieved in these soils. So to summarize that sodic saline presentation, just from the management point of view, from the last few slides, I think the deep place of organic amendments and gypsum significantly can increase the yield. We observe the better root growth. We also improve the infiltration rate and crop water use. The mechanisms, as I explained, pH of highly alkaline soils needs to be reduced, and that is a mechanism that we always suggest gypsum is operating. It's not just about calcium, but it's about the pH effect of gypsum. Organic carbon in soil fraction improved up to 4-fold improvements in the soil structure. So if it's a multiple soil constraint, if you have dispersion, sodicity, and a poor structure, you need gypsum for the dispersion bit, you need carbon for the structure. So the message is not just a simple gypsum application alone is going to fix all the problem. The message is you need a bit of carbon in the system in form of amendments in this case to improve that structure. This was my presentation, Felicity, about sodic dispersive soils. It's 10:15. What should we do? Ask
yes, yeah, we might have to
Can't
move
hear you.
on, SN, unfortunately. There was a question about gypsum. So Tim has asked, what determines how long a gypsum application will last in reducing the impacts of a sodic soil?
Okay, that's easier to answer about how long lime will last in an acidic soil than gypsum. To be honest, I think there are two issues here. If you're talking about the surface soil application, and that means you have a surface soil dispersion, basically the best answer to that is the crop performance. If you start to see a decline in your crop establishment or crop performance, then that is the time that you need to apply gypsum again. In terms of the subsoil constraints, is a very difficult story because there is nothing that can actually measure that when gypsum needs to be added again. But I would say surface crusting, crop germination, and crop overall health at the early time of the crop growing season is the best determination of when gypsum needs to be applied again. And it's in a few years' time. It's not something that we need to add every year.
Yeah. And just a question about the economics of this work that you've been doing, Eshan. I know that it's work that can be done by a research organisation because we can invest in that area. Is it something that can be, is it economically viable to be adopted in a broad acre sense? And did you crunch any numbers in the cost per hectare in this work?
Yeah, look, it's a question that I get all the time. We get all the time as a team. And yes, first of all, I give you a number of examples. The rainfall or irrigation is the number one factor that is dictating whether you need to do soil management or not. If we are in a low rainfall area, doing some of the example that I gave in soil management is probably not viable and feasible because you need to have water in the system to drive the interaction. And for me, soil constraint is where you have unused water in the subsoil and the crop can see that because of the soil constraint. If there is no subsoil water, there is then, and you have still a subsoil constraint, there is not a lot of, the risk is high because you are not actually going to improve the system by much, even though if you are fixed the soil, there is no water. So for a low rainfall area, I recommend use of genetic tolerance is the best option. Let's target the best variety that we can use in broadacre cropping system that are tolerant into those systems. If you are in a medium and high rainfall area, then yes. Almost all the farmers that I'm working with in the southern New South Wales and a number of other farmers in high rainfall Victoria, they are adopted this work commercially and they are working on expanding this work in their business. The reason is that they've seen quite a remarkable transformation, and I think the soil structure that I brought the example is from a high rainfall area in Victoria where the farmer now having their own commercial machinery for this type of work. I still think machinery is the bottleneck, Felicity. We need to improve the way that we are incorporating our amendments into the subsoil, and there's a lot of work is being done as we speak. The amendment is very also important because if you need to pay for the freight, is the killer. Is the most of the cost is coming from the freight. So that's why in my own research we are going through developing a platform where we can use farm-grown biomass, such as wheat stubble or crop biomass, as a source of carbon to be actually returned into the subsoil. And it's not black and white to say whether it's feasible or not. I really think we need up to 5-7 years of data to be able to say, okay, this is the initial cost that happen to do with this management, and this is the residual effect of a one-off application of organic or inorganic amendments. And then that whole 5-7 years of data will develop a business case or economic analysis. So far, it has been quite encouraging. Very, very simple. If I want to put a number into the paper, our economist tells us if we can find an extra ton per hectare after the soil management on top of, it's actually going to be very viable, and we are achieving that at the moment. So you will be unlucky if you hit by drought immediately after you manage your soils, because and we experience that. But yeah, so far I'm really positive about the potential benefits of this in medium and high rainfall areas.
Yeah, and I think, like you say, it's that it's the benefits over time. So you can't just take it into account for 1 year. That hope, like
it is not. No. For 1 year, it is not feasible. That's right.
Yeah. Anne's asked about a couple of products. Have you used or know of a product called Soil Life Organic Biocatalyst and Biostimulants? Have you used any of those products?
I haven't used any of those products. And the biostimulants, I don't think is a product. I think it's a more broader group of products that's showing a lot of interest. It's huge in America. I know that. And I think in long term, if it's actually tested in Australian farming systems, there might be some potential benefits. But I personally, I used either the existing products or the most cost-effective products for a source of organic matter. However, as I mentioned, we are developing new formulation of products that are going to target sodic or acidic soils. So that's the new generation of products as part of CRC program that is underway.
Terrific. Thank you, SN. Apologies that we've run out of time. We might need to hold up the second part of
yes, definitely. Yeah.
To another day, but that's okay. The Southern group will get to hear it all tomorrow, so you'll get to talk about it tomorrow, and yeah, we can attack a soil acidity at another time. But thank you very much. If you have any other questions for SN, are you able just to hang on the line while Moran gives his talk? And some people
sure.
Chat, pop some questions. If you have a look at the questions in the chat, SN, and if you've got anything more to add, then you can pop it in the chat. But thank you very much for making yourself available. I would now like to introduce Moran from Griffith University to talk about his topic, which is about sorry, Moran, perhaps you'd like to introduce yourself.
That's all right.
A bit of a hash of it. You'll do a much better job than me.
It's a bit complicated, I understand.
It is.
Okay, let me try to share my screen. It's getting complicated.
There it is.
Good. So can you see it properly?
No, so we can see the two screens. So if you just change the display settings to show one screen.
I think that's the better way they can do that. Because I put it on full screen, it goes to the other side. So is it fine now?
If you go down to the, yeah.
If I put it on a slideshow, it becomes like this.
Okay. Yeah. Okay. If you go up to display settings, does it? If you go up to display settings up the top.
Meran?
Yep.
If you put it on a slideshow again, please.
It is on a slideshow.
Yep,
right now.
it's very easy to fix actually.
But it goes to my left-hand side.
On the top panel there, exactly. If you go up, display setting in the middle, up, yep, display setting, yep.
Okay.
Yep, and swap, yep, swap presenter, yep.
Perfect. Thank you, Esa.
Thanks, Esan.
Thank you. So, can you see it properly now? Can you hear me as well?
Yes. Thank you, Meran.
Good. So, I'm just trying to make a short presentation about this project that we started to work with Soil CRC. The idea behind this project was that, a lot of different groups and people measuring soil health. So we have many different parameters to measure soil health, but the problem is that the results are usually not consistent, and it's really difficult to adopt it in farming systems because they become so expensive. So in collaboration with Soil CRC, we end up with an idea of evaluating soil functional resilience to environmental stresses, in this case, the compaction and drought, which are two of the main environmental stresses in Australia. We wanted to have a mechanism-based understanding of how these stresses affect on soil health, and eventually, hopefully by the end of this project, be able to reduce the number of the tests that we have to do and reduce the cost of sampling and analysis of soil to end up with a simple, robust, and applicable soil health package test for different farming systems to be able to adopt it in their own farming system, their own paddocks, with minimum cost and maximum, basically, value. So we end up with a project, we call it Evaluating Soil Functional Resilience to Compaction and Drought Stresses for Developing Higher Performance Soils. So in these projects, we have researchers from Griffith University, New South Wales DPI, and we also work with two farming groups. One, Herbert Kane Productivity Services from Queensland, which I see Richard is representing them here. And also the FACEI group from Western Australia. So the FACEI groups mainly deal with the drought problems, and basically Queensland farmers mainly have issues with compactions. So we try to put them both together and have a proper mechanism-based understanding of how the soil reacts to environmental stresses to be able to end up with some solutions. So some background before I show you some findings of the projects. As you know, the definition of a healthy soil is a soil that have the continuously capacity to provide optimum amounts of nutrients, water, and air for growing plants and provide a disease-free environment for the cropping systems. But due to over-application of mineral fertilizers and conventional management systems that adopted to different farming systems, we end up with a continuous decline of the yield and economic profitability of the farming systems, which mainly associated with reduction in soil fertility and health due to different environmental stresses and basically adopting of different farming systems. But if you want to make a change, first we need to end up with a simple, robust, and affordable method for farmers to test their health status and give them basically farm-based or paddock-based recommendation to be able to improve the health of their soil and improve the fertility of the soil. In order to do that, we first have to have a good understanding of the process that the soil goes through when they're dealing with the environmental stresses. So if you look at the graph here, I'm trying to simplify what happens when the soil goes through different environmental stresses. So they have two definitions here. The first one is soil resistance, and the second one, soil resilience. So for example, if you're at the top of the graph, your normal soil in the paddock, when you're dealing with a different environmental stress, such as drought and compaction, the functionality of soil, physical, chemical, and biological properties start to degrade. So the capacity of the soil to deal with the environmental stresses is called the resistance. So the soil start to resist against the changes that force through the system by different environmental stresses. But what happens if the environmental stresses becomes long-term or so frequent to happen? Then the soil cannot basically resist against the changes. So the soil have to change. Physical properties, chemical properties, and mainly biological properties start to change when the stresses become more frequent or so long-term. So as you see, the basically functionality of the soil start to decrease when we're dealing with these kind of stresses. But what happens when the stress is lifted, when we remove the stresses, when we do plugging, if we have the compaction problem, when we irrigate the soil, when we have drought problem, or we have a rainfall. So the soil start to restore its functionality, physical, chemical, and biological. The capacity of the soil to recover itself and get as close as possible to its original functionality is called resilient, which is the basically main point of these projects that we try to improve. If we end up to improving soil resilient, we can make sure that when we have environmental stresses in our systems and the environmental stress is lifted after a while, we have a higher chance to recover our soil functionality to its close to its optimal condition. Why is it important? Because as you see in the graph, every time that your soil goes through different environmental stresses, lose parts of its functionality. And when it recovers, it doesn't recover to its optimum condition. So it's lose its functionality after each time. If this process happens over a really long time or more frequent over a long time, eventually you end up to a status that the soil will irreversibly damage or permanently damage. So the functionality will go so low that it cannot recover itself. And that's the point. Then we started to get soil degradation. We started to lose the profitability. We start to lose the crop yield. And the cropping system is not profitable for farmers. The problem here is that if the proper profitability goes down, then our capacity to improve soil health by applying different amendment goes down, because there is no profit in the system to be able to use to improve soil health. So we don't want to get to that critical threshold point. Before we get to that point, we need to do something to the soil as long as we have enough profitability to reinvest into the soil system. So these projects try to have an optimum understanding of the mechanism-based processes that happens in soil microbial community composition and diversity, soil chemical properties, when the soils go through drought and compaction stresses across different farming systems, and end up with a model that can predict soil health status, and then simplify that model to a couple of simple tests and couple of cheap tests that's affordable for every farmers, every farming systems to test their soil health and improve it. So the expected outcomes of these projects is broad application of the better management practices through different projects that going through Soil CRC, different farm-based projects, and also for a broader Australian agriculture industries. Here is an example of how we do our incubation experiments. Before I go further, I need to mention that these experiments are our lab-based experiments, because the idea of behind these projects just to find a mechanism-based understanding of the process to help us to get our finding to the farming lands and to the broad acre cropping systems. So the experiments that we do here are all lab-based experiments. For example, you see here, we have a jar that we put the soil in, and we use our compactor machine to compact the soil to different compaction systems. We also introduce different routes to the samples, and we look at their functionality. So these projects try to develop different assessment methods and management protocols to improve soil functional resilience against the stresses. Why is it important? For example, if you look at the graphs on the right-hand side, when you have a resilient soil, you have reduced the amounts of your carbon loss, either through leaching from the subsoil or through basically CO2 emission or greenhouse gas emissions. And also you can keep the integrity of the soil intact. Why is it important? Because in a good year that you have enough rainfall or a good condition in your farming systems, basically most of our soils produce similar amount of yield. But in a bad year that you don't have enough rainfall in the system, the soil is not in its optimum condition. A resilient soil can guarantee a minimum yield and can guarantee that you don't have too much loss in your profitability, and you can safely basically through those bad years and move on to the next couple of years, which probably you get a better condition by changing the environmental processes. So how we do that? We provide different stresses to the soil, and we assess soil functional processes and microbial community composition and diversity, and relate them to the soil fertility and plant productivity. We also try to provide practical methods using innovative biochemical methods for re-engineering of target soils against these different environmental stresses, and also providing soil resilient protocols to the other project leaders of Soil CRC to be able to test it in different cropping systems and basically modify it based on the requirement of the different cropping systems. Also, we do international collaboration through basically providing different papers, journal papers, or presentation in different conferences. So what we have done till here, we had different meetings with the research providers and the farmers community and farming cropping systems to design different experiments. We selected different field sites, both in Western Australia and North Queensland, under different gradient of management practices for our experiments. We're in the process of providing a literature review paper, which will be useful both in Australia and worldwide to have a better understanding of the resilience of the soil against different environmental stresses, which is not a really simple concept when you bring it to the broad acre systems, and also providing different scientific papers, technical reports, fact sheets. But just remember, this is a lab-based experiment, and we're not applying anything to the broad acre field systems at this stage. So what happened till now? We finished a literature review of the projects. The PhD students that has been involved in these projects had a thorough literature review regarding the effect of soil compaction and drought on basically soil health and improving the resilience of soil when they're dealing with these problems. We finished the first experiments of the project, which was basically 72 days incubation experiment. So for this experiment, we picked two soil types, a clay soil and a sandy soil, two levels of soil moisture content, 15% and 35% water-filled porous space. Just need to remind you that water-filled porous space is different from water holding capacity. When we're talking about water-filled porous space, we're mainly talking about the amount of the soil pores that filled with water. So it's different from how much water can be held in the soil. It's mainly a combination of the compaction and availability of water. So when you put them together, it becomes a different factor. We call it water-filled porous space, which means how much of the porosity is filled with water, which decide if your system is aerobic or anaerobic, which really helps in interpreting your result, because mainly when you're talking about the biological systems and soil microbial activity, it's really important to know your systems is aerobic or anaerobic and what kind of organisms are active in each condition. We also used 6 different levels of compaction, from a really low compaction or 0.9 gram per cubic centimeter up to 1.5 gram per cubic centimeters, which is a severe compaction issue in the farming system. So we tried to identify the main indicator of soil resilience. How we did that? We measured a combination of different chemical and biological properties. Also measured the diversity of soil microbial communities to be able to identify the most important factors and have an understanding of the mechanisms that are going around, to be able to later on simplify the process and also introduce the application of organic matter and see the effect of the system or change on the system when you introduce an organic matter. So the first incubation study was based on understanding of the mechanisms without improving soil condition by application of organic matters. So the findings for these experiments indicated that soil microbial metabolic activity, as measured by the respiration or enzyme activity, responded differently to different levels of the drought and compaction, and also were really different on different soil textures. So to have a good understanding of what I mean by microbial metabolic activity, I can explain that, for example, assume that your soil profile is living like a human. So for example, the soil profile is living like me. So generally, I respire more CO2 than you guys because I'm fatter, I'm heavier, you know, I have more body mass. So when I have more body mass, it means that my metabolism is faster, my metabolism is higher, so which means that I produce more CO2. The other reason that I can produce more CO2 is when I'm running, when I'm active, when I'm doing something. So my body is under stress, so my metabolism goes higher. So although I have the same body mass, but I'm respiring more CO2 because I'm under stress. So what happens here, we try to differentiate between the amount of enzyme activities and CO2 respiration into the soil system when the soil is healthy, and the reason for more enzyme activities and CO2 respiration is because the soil is producing more microorganisms and we have more mass and we produce more CO2. Or when we produce more CO2, the reason is because the soil is under stress, our microbial community are fighting for their life and try to generate more CO2 because they're more active. So what we have done, we had our two different soil types, a clay soil and a sandy soil. We put the soils under different stresses of compaction and drought. The first step of the experiment was to apply the stress. As you can see here, we applied the stress at the beginning of the experiment, and we monitored the changes in soil chemical and biological properties to see how they degrade when they get affected by environmental stresses. For example, here, the graphs that you're watching is related to the extraction of hot water extractable organic carbon. This is slightly different from extraction of organic carbon. In this case, we keep the soils in hot water, 70 degrees centigrade for around 16 hours, and then we extract the samples. Our previous experiment shows that this is higher relationship between the extracted hot water extraction of organic carbon and your available food for microbial activities. So when you're measuring hot water extractable organic carbon, you have a better understanding of how much food is available for your microbial activities to grow. As you see, the pattern is slightly different when you're comparing a sandy soil to a clay soil, and also when you're comparing a high moisture condition to a low moisture condition. But overall, in all of the graphs, when you put the soil under stress, your food availability or hot water extractable organic carbon availability decreased by time. When we left the samples for 35 days, we decided to release the stress. So if it was under drought stress, we add water to the system. If it was under compaction, we remove the compaction and to see how microbial community react. So for example, in this case, we release the compaction and add water to the systems, and you see the hot water extractable organic carbon start to increase. So the food availability start to increase in the systems, means that the composition of organic matter is accelerating, but you see there is a gap between different compaction levels, and there is a gap between recovery of the soil systems after the releasing from the stress. So the process is different from sandy to clay soil, and the process is different from low moisture condition to high moisture condition. Similar things happens when we look at the respiration of different soils in different condition. As you can see, when the soils start to go under stress at the beginning of the experiment, the difference between treatments minimized because all of them have been under stress, all of them been reducing their functionality. So the CO2 respiration decreased as a result of reducing the number of microorganisms. But when you release the compaction stress, they completely react differently. Some of them recover really fast. In this case, the one with the low compaction. So, and basically higher moisture content, especially you can see here, if you have high moisture content in a clay soil, the recovery rate will be completely different comparing to the time that you have low moisture content. And the same happens if you're looking at a sandy soils, the recovery rate is completely different. So this will give us some idea of how we can basically relate different analysis systems, which are really cheap. For example, here measuring CO2 emission is really cheap. So you see that we can capture the difference between recovery of the soils using a simple test like CO2 emission. But the trick is here that we need to relate this simple test to different properties of the soil, biological property, chemical property, and physical properties to make sure that the result that we're getting is a true representative of the soil. And that's why we measure a lot more different parameters to find out if a result are robust or explainable or not. For example, here we're measuring one of the main enzymes that responsible for external digestion of the food. You know that the microorganisms cannot ingest the food inside their body. They need to digest the food outside and then basically consume the digested food. One of these enzymes, which is external exudation of soil microbial communities, is beta-glucodase, which is effective in soil carbon cycles and effective in the composition and release of carbon in the soil systems. You see, we have the same pattern. When the soil goes under stress, the exudation of beta-glucodase decrease. And when we release the compaction, we have more enzyme activities in the systems. And these enzyme activities are different when we're dealing with different compaction levels and different moisture levels, and also in different soil types and soil textures. Next one, the changes in soil microbial community composition. So if you look at the left-hand side in A, that's our soil that has been under no stress, so it's optimum condition. And in the left-hand side, you see a soil that has been under compaction and drought stress for 35 days. And you see that the community composition of soil microbes are changing, and it means that the functions of soils are changing when it goes through different environmental stresses. One example, which is a really simple example, if you put your clay soils, which is really common in North Queensland, when you put your clay soil under compaction stress, you reduce the amount of CO2 that's produced, comparing the soil that has been under no stress. So it means that the microbial community comes down, less CO2 production in the systems. But because the systems change from aerobic condition to anaerobic condition, the anaerobic microorganisms start to increase, and your nitrous oxide emission increased by 5-fold. And this is one of the other issues that we have in North Queensland, which we lose a lot of nitrogen through greenhouse gas emission, similar to runoff, similar to deep drainage. But the changes in the compaction not only affect the carbon cycles, but also affect the nitrogen cycles and nitrogen use efficiency. And that's related to the change of microbial community composition in aerobic and anaerobic species. After we finished that experiment, we went to the second phase of the project, which in this case, now we understand the limitations that we have in our cropping systems. We know how the systems works when it goes under stress, and we know how the system reacts when it gets out of the stress. Now, in the second part of the project, we try to introduce crop residues to the systems and find out how the crop residues will affect and improve soil resilience against drought and compaction. So this is the part of the projects which is already in progress. So for example, we designed 2 different experiments. One experiment was mainly focused on drought, and the other experiment was mainly focused on compaction. So this experiment you see here is the drought part of the experiment. We have 12 treatments, and we collected 2 different soils adjunct to each other. So they have the soil structure, the same soil texture, and basically the same nutrient profile, but they have been under different management practices. So the topsoil has been under conventional management practice for a couple of years, and the bottom soil from treatment 7 has been under best management practice for a couple of years. So we have 2 different soil types, two different soil adjacent to each other, same texture, different management practices. The same as previous experiment, we put them under stress and to see how their functional properties change when they go under stress. Is the historical management practice has any effect on changing the mechanism when the soil go under stress? And then after a while, we remove the stress. For example, in this case, we remove the drought And also we apply organic matter content. So here you can see when we lift the drought, we had 3 moisture condition, 15% water holding capacity, 30% water holding capacity, and 55% water holding capacity at the beginning of the experiment. But when we lift the drought, we remove them, we bring them up, all of them to 55% water holding capacity, which is the optimum condition for plant growth. And also for half of the treatment, we apply the organic matter into the system. So this organic matter has been chosen based on the available organic matter in different farming systems. We apply to the systems, and the same process, we look at the mechanisms of the recovery of different basically treatments without application of organic matter, and the one that has been under stress, now recovered, comparing the one that hasn't been under stress into this system. And that's it. So I'm happy to take any questions.
Thank you, Miran. That was a great summary of the work that you've been doing. The group has, it's quite complimentary for the group because they've had a talk on soil biology, so they can understand what you've been talking about in regards to those stresses and the impact on the soil biological activity in the soil. I haven't got any questions in the chat, but if anybody has any questions, feel free to unmute yourself and fire away at Miran. I know that some of the group are interested in measuring soil biological activity and have been using or are looking at using a microbiometer. Do you have any opinion on microbiometers?
So when you're looking at the microbial activities, you need to separate their activity based on the optimum condition and their stress. So sometimes you have higher microbial activities. If you're looking at their mass, it's a good representative of their activity. So when they have higher mass, they have higher activities. But measuring the mass is usually more problematic and more expensive. So the easiest way is to see the respiration. But the problem with the respiration is that you need to differentiate to see if this respiration is coming from the optimum condition or it's coming from a stress condition. So any products available commercially can be easily calibrated against the standard methods. And when you calibrate those products, either it's for measuring the microbial activity or measuring the respiration, when you calibrate it properly based on the soil type and basically the methods that we use in the labs, and you calculate the amount of error, then you can accurately use them in your farming systems. Really cheap, really effective, but already calibrated to the optimum condition.
Yeah, thanks. Any other questions yet, Brendan? Have you?
Yeah, I was just, did I miss this, Miran? But is this going to be a commercial test of which we can send samples in? Sorry, how's this working? So if we had, say, a trial where we're looking to apply amendments, can we send samples to you as a commercial entity to get tested? Is that how this is going to be set up?
Yeah, eventually it will be something like that, but I don't think Griffiths will be able to expand it commercially because it will be an IP for Soil CRC, and
Yep.
eventually the Soil CRC will decide how does it want to commercialize it. So basically, what we promise Soil CRC is that we do a switch source of a lot of different parameters, which is really expensive. But we try to connect them together. And then when we get the connection between these parameters, then we can eliminate a lot of them and get the relationship between them. So we end up with a really simple test, but has a basically mechanism-based proof that it works. So when it gets simplified like that, it will become like an IP. And then eventually Soil CRC will decide how to use that IP. But definitely for the farming systems that are part of soil CRC, it should come like a free of charge and as a part of the other projects, because this will be part of the other projects that going to happen in the next couple of years in soil CRC. So they're going to use this test in the farming, different farming system, and to see how they react. And basically you need to modify that based on your farming system as well. So there are some tricks to modify that, but eventually it will become a really cheap commercial things that would be available to everyone. But how it becomes available, I don't think we can make a decision about that. Eventually soil CRC has to make a decision about that.
Thanks, Maram. I see SN still on, so if anyone has any other questions for SN as well, please feel free to unmute yourself and fire away.
Yeah, I had another question, Hassan.
Yeah, sure.
When we talk about deep application of soil amendments, I guess we're talking in that sort of, 25 to 30 centimetre depth zone. Our concern is if we go and place products at that depth, are we just creating a lovely little environment down there, but effectively the soil reconsolidates above it? So effectively roots or the rooting system of the plants then can't access it, because over time, the soil above that treated area then reconsolidates and still can't gain access to what we've just applied at that 30 centimetre depth. So is it a mechanical thing rather than just placing product at this lovely little pocket, or should we be treating the whole profile, like wicking it through the whole profile, and mechanically, how do we do that?
Yeah, so before I answer your question, you're talking about broadacre cropping?
Absolutely, dry land, broadacre cropping.
Yeah, and what issue are you talking about? Salinity, sodicity, or acidity problem?
Probably more sodicity.
Sodicity, yeah, okay. Because the question is really good, if you have acidity issue, it's low soil pH from 5 or 7 centimeter all the way to 20, 30, the best thing is to treat the whole profile, okay? If you don't treat that acidic layer, the crop response is going to be minimal, because you still leave behind a lot of aluminum toxicity in the soil. In terms of sodicity, we are dealing with the soil structure. And what I can tell you, when you're putting the amendments at depths of 20, 30, 40 centimeter, with our machinery, by the way, it has a dual depth delivery. So the 2 times when they're going into the subsoil, a band of amendment goes at about 15 centimeter. The second band goes at about 35 centimeter. So you cover quite a bit of depth, but based on diffusion of the carbon that I talked about. So you have organic amendments, and then it's going to break down, and you have mineralization of carbon, and that carbon will diffuse into the surrounding area and will improve the soil further. Of course, the best way is to put the amendments all the way, but you need to identify if there is a problem to put the amendment there. The top 10, 20 centimeter often is well structured, or better structured, if you like to put it that way. It's not a mechanical thing, because my observation is that when we are doing the deep ripping alone, if you don't have compaction, the deep ripping alone is not actually giving us any yield benefit whatsoever. It's only important if you have a compacted layer. Otherwise, it's about the chemistry and the interaction of that amendment that is improving that hostile condition. And no, based on what we are actually seeing, a one-off application is lasting 5, 7, up to 10 years. So I don't think that above layer, the 20 centimeter, is going to give us any issue in the future. And imagine a wheat crop or even a canola crop goes up to a meter 20, at least in my part of the world. But yeah, it's more about the profile below that layer. So if I put the amendments about 20, 40 centimeter, I like to see, now that we improve that lovely area that you mentioned, can that be a way for the root to penetrate into deeper soil profile more vigorously and more strongly. That is what I want to achieve.
Thank you, Ehsan. I guess thanks, Brendan, for your question, and thanks to the others who have posed questions for our speakers. I'm conscious that we've gone over our time, so I don't want to hold you guys up, and very appreciative again, Ehsan and Maran, for your time and your expertise in sharing with these guys, and I'm sure you'd be happy to continue any dialogue with the community of practice if you guys have any further questions for Ehsan and Maran, then I'm sure they're happy to address those. So thanks very much everybody for participating, and it's nice to see you all fit and healthy. So in these funny times, please stay well, and I'll be in touch with the next times we can catch up with each other. So, but I'll hang on the line if anyone wants to ask me any questions or discuss anything after this meeting.
Before you go, Felicity, so
yeah.
Tomorrow for the Southern panel is at the same time?
Yeah, that's right.
Yeah, okay, sure.
Yeah, thanks. All right. Thanks, guys. Thanks
Thank
very
you.
much. Nice to see you all.